Hi guys, I'm gonna see if I can get through a little more of this tonight and see if we can kind of catch back up a little bit. I know we're a little bit behind. Um, anyway, I'm gonna try to get through with the respiratory system tonight. I think I've digested already, recorded from um, the first part of the COVID outbreak back in March. I think my A&P2 class that I worked with then, I recorded a couple lectures on Loom. I'm going to look them up. If not, I'm going to have to do about 20 hours of lecture to finish out the semester. I hate doing these. It takes a lot of my time, especially when I'm teaching during the day in lab and having to do these too. Anyway, we'll work our way around it. At least they'll be recorded maybe for next time in case something else happens and have to deal with it. Anyway, oxygen and CO2. We're going to look at how they're transported in the blood. Oxygen is pretty simple. Oxygen basically binds to hemoglobin in your red blood cells, and it's carried basically on the heme group. And there's 4 oxygen molecules for every heme molecule. But that really means, really there's millions and billions of oxygen molecules carried throughout your blood at any given time. And we'll look at what it means to only have 4 locked to the heme group of hemoglobin and why that's important. Most of the time we only dump one of those off and we keep the other 3. And there's a really important factor to that, and we'll get into it. Molecular oxygen, which is O2, I don't know if you remember back from A&P1, the chemistry section. Oxygen is one of those diatomic molecules that in nature is always found as a diatomic molecule where 2 oxygen atoms are bound to each other. So we always have to put it as O2. Nitrogen's one, chlorine's another, iodide. There's 7 of them. Hydrogen. All of those are going to bind with themselves with a covalent bond and share electrons. But oxygen in our atmosphere is going to always be found as O2, and we're going to have to find a way to bind that to a molecule in our system. And the problem with oxygen is it's very, very reactive, and oxygen's not good for us. And believe it or not, even though we have to breathe it to survive and we'll die without it, initially oxygen was very toxic to most living organisms, and it still is. And when you get in microbiology, you'll start to understand a little more about that when you deal with anaerobic bacteria and how they can't tolerate oxygen. It's because of the oxidative factor. Just think of an apple. When you bite into it and you leave it sitting for a minute, it turns brown. That's the oxidation of those cells in that apple. Same thing happens in us. So our body, over time, and every other oxygen-breathing organism, had to accommodate that oxygen somehow by taking it and making it less toxic. And we did that through our metabolism and our metabolic pathways. And some bacteria did that to begin with, and we think, on and on, that got passed out. But most of the oxygen we carry is going to be bound to hemoglobin. And what that means is when it's bound to hemoglobin, it can't react to anything else. Until we get ready to deliver that oxygen to the cell to where we can get rid of it and burn it up through cellular metabolism, it's inert, and it's not going to be able to react with our tissues and things. And again, we don't want that. Just leave a piece of metal out and put some water on it and let oxygen interact with it, and it will turn to rust. Your body will do the same thing. But again, we have developed metabolic pathways to get around that. So there's oxygen molecules per hemoglobin molecule, and there's a little bit dissolved in the plasma. I mean, 1.5% is very negligible. So 98.5% is bound to the iron of hemoglobin. So if you have iron deficiency anemia, that's why you are anemic. You're not getting enough oxygen because you don't have enough iron. Anemia is going to be a term, again, that's a catch-all term for any time you can't carry enough oxygen in the blood. It can be due to not having iron, or it can be due to other things like pernicious anemia, where you don't produce intrinsic factor in your intestinal tract. And we'll get to that later on too. Um, so anyway, oxyhemoglobin is when we have hemoglobin bound to oxygen. This is a reversible reaction. So in the lungs, we add oxygen to hemoglobin. In the tissues, we pull it away from it. And basically, we go through an oxidation reduction reaction. And it's a very simple reaction, but it's a very important reaction. In that we get oxygen from the blood to our cells so that they can carry out cellular metabolism. So loading and unloading of oxygen is facilitated by the shape change of hemoglobin. As more oxygen binds to hemoglobin, it wants more oxygen. It's almost like a drug addict or something. Once it gets a taste of oxygen, it wants more and more and more and more until it fills those 4 zones that it can fill. As it's released, oxygen becomes less, attractive to hemoglobin, and it wants to start picking up a little bit of CO2. So when it's partially saturated, 1 to 3 hemes can carry oxygen, but the other can carry some CO2. And we'll look at that in a minute. But normally, hemoglobin wants to carry those 4 and be 100% saturated, if at all possible. Sometimes that's not. If our lungs are not functioning properly or our bloodstream is flowing too slow or too fast, we may not be able to do that. Or if we have some kind of obstruction, not letting enough oxygen in it, we may not be able to do that. But most times in a normal situation, 4 heme groups are going to be bound to oxygen, and we're going to hold those there. So the rate of oxygen is regulated by, number one, partial pressure. Remember, that's the big thing with oxygen is the partial pressure. It's not very soluble, but it has a big concentration gradient between how much partial pressure we have on this side of the membrane versus this side of the membrane in the alveoli. Temperature is also going to play a role with this. And this may just sound strange as it can be, but I went trout fishing in Montana a few weeks ago. The reason trout live there, reproduce there, most of those trout are not native there. There's only one species that is, and the rest of them are introduced, but they took over and live in that river because the oxygen level is very high and the temperature is very low. If that temperature goes up just a few degrees, they start dying off or moving to other parts of the river or down into a lake or somewhere else to where they can reproduce. So it's one of those things where temperature and oxygen levels can really vary in the blood or even in water. Any kind of liquid like that, the higher the temperature goes, the less soluble gases get in that liquid. It's absolutely the opposite of what a solid would be. You think of sweet tea and you're dissolving sugar in sweet tea. The hotter you get that mixture, the more sugar will dissolve in it, or the more solid will dissolve in it. But with gases, it's totally the opposite. The lower the temperature of the liquid, the more oxygen it can contain. So again, temperature is going to be one of those things. pH is another one. That really plays a big role in the shape of the hemoglobin molecule. And if it gets too far out of range, a hemoglobin molecule will shift its shape and it won't hold as much oxygen as it normally can. CO2 concentrations also play a role, so the partial pressure of CO2. And then the concentration of bisphosphoglycerate in the blood, which is a byproduct of cellular metabolism. So during glycolysis, during the anaerobic phase of metabolism by red blood cells, this is also going to affect the amount of oxygen that can bind to hemoglobin. There's a lot of things that can make oxygen not want to bind to hemoglobin and not want to be carried in the blood effectively. And we have to look at all of these. So let's say you get a patient and they're low on oxygen, high on CO2. You got to look at what's causing it. It could be a lot of different factors. It could be their lungs are not getting enough oxygen to the blood. It could be they may have a fever or a temperature change. Their pH, and maybe an acidosis, alkalosis. CO2 could be leading to acidosis if they have too much of it. And then the red blood cells may be basically going through glycolysis and not getting into cellular respiration, not going through the other cycles to where they can get rid of some of these byproducts. So there's a lot of different things that can go on with this. So oxygen hemoglobin dissociation curve, we'll look at this on the graph, and I'd rather look at it than explain it. But if you look at what we have going on with the percent oxygen saturation of hemoglobin and then the partial pressure of oxygen, if we have 100 here, then we basically can go up and saturate everything. And this goes back to that 25%, 50%, 75%, 100% saturation we looked at in the other curve in the last video. If you look, we are here at 25%. So if I'm exercising, I am only loading back 25% of the oxygen that I could be to the hemoglobin, right? If I am resting, I am putting back almost 75% of the hemoglobin, right? And then in the lungs, I'm actually trying to do 100%. So I'm trying to get that 100% saturation. But what's going to happen is it leaves the lungs and goes through the tissues. We start coming back down here. We start using that oxygen. So when we're exercising, we take off a good bit of the hemoglobin, 75% of it, and we're only left with 25%. If we're resting, we're left with 75%. And I told you in the last video this is going to be important. The reason you do CPR the way you do nowadays, they really just want you to do questions. They're not even worried about, especially with COVID, they're not worried about you getting breath into that person. All they want to do is get that blood moving around, right? So let's say a person is laying on the floor and they're not breathing, you don't feel pulse. You start pumping blood. Well, if they were exercising and fall out, well, there may not be a whole lot you can do for them. But if they just fall out and they're at rest, they still have 75% of the oxygen on the hemoglobin that they had before they fell out. So what can you do? You can pump that heart, and you still have 70% or 75% saturation, and you can go around and unload at least 2 more times to the cells, namely of the brain, and get that brain tissue some oxygen so that it can survive. So when you have that extra amount of oxygen bound to hemoglobin, and you're only taking 25% off at any given time. So if I'm at rest, I'm still saying it at 75% saturation. I can sit here and pump your heart and move that blood through your body without you actually breathing and still give oxygen to those cells and still get those cells the nourishment and oxygen they need until someone can get there to revive them. Again, if you've taken CPR, you know your goal is not to bring a person back to life. It's just to keep them alive until someone can basically get there that can do more than you can do. Right. So what we need to look at is the influence of partial pressure of oxygen on hemoglobin saturation. So in arterial blood, that's freshly oxygenated blood that's being pumped out of the heart. The partial pressure of oxygen is 100 millimeters of mercury. It contains about 20 milliliters of oxygen per 20 milliliters of blood. So you think back to what we talked about, the concentration of oxygen in the atmosphere being, that's 21%. This is almost 100% of the oxygen you breathe in getting into your system if your body is working properly. So your hemoglobin is 98% saturated at all times. Again, that's assuming you have a healthy lung, and that means your lungs are functioning normally and doing what they're supposed to do. Now, further increases in oxygen, so let's say I breathe deep or I even take in oxygen, I'm not going to saturate hemoglobin anymore. The problem is, though, if you give someone oxygen when they're already saturated, where's that oxygen going to go? Now we are increasing the partial pressure of oxygen even more than it is normally. Let's say I'm breathing in 80% oxygen instead of 21%. Now I am perfusing the blood with more oxygen than normal. Now, if I have a non-functional lung, that's great stuff because now I can match up what I should be putting in the blood with my disorder. So let's say my lung capacity is down by 20%. Well, now I'm not getting as much oxygen in my blood as I normally would. So I up the oxygen concentration, and that puts more oxygen into the bloodstream. If you're healthy, though, and you're already saturating at 98% and you breathe oxygen, now we start to get oxygen that wants to dissolve into the plasma. And when that happens, it's just like the apple. It starts wanting to oxidize things in the body. There was a big craze back in the 90s, things called oxygen bars, where people would go in and breathe flavored oxygen. It was cherry and grape and strawberry, whatever you wanted. And it made you feel a little bit better because it kind of gave you a boost, especially in casinos and things where people would stay up all night, play cards or whatever until 4 or 5 o'clock in the morning, and then they woke up at 7 or 8 and were hungover. They could go to the oxygen bar and get a little jolt of energy and then start playing cards again. That's what the casino wanted. And same thing happened with bars in the bar districts like Buckhead back in the day and stuff. That people would go up there, drink all night or whatever, and they'd wake up in the morning and they'd be like, hey, I got to go to work, I got to do this, and they would go to the oxygen bar for a little while and rejuvenate themselves. But what they found when they were doing that was that they were doing a lot of cell damage because most of those people were healthy and their lungs were working properly, and they were getting too much oxygen into the bloodstream. So again, lesson from that, like anything else, too much of anything can be bad. So in the venous blood, the oxygen partial pressure is pretty low. It contains about 15% oxygen, but hemoglobin is still 75% saturated. So again, in a normal cycle, we only pull one oxygen molecule off the hemoglobin per round that it goes out through the body. So theoretically, again, even if you're not breathing, you've got at least 3 cycles that your body can go through and still oxygenate cells before you die. And that's a good thing for us. And again, that's why CPR works. It pushes that blood around with that oxygen in it to get it to vulnerable tissues like the brain, to make sure that they are not deprived of oxygen. The heart, same way. We don't want it deprived of oxygen. And hopefully, by doing that, you can keep someone in a good zone before paramedics get there and they can get hospital care or whatever they need. I'm going to skip these and just kind of head to the curve again. So again, this is just what the chemistry of hemoglobin does. It saturates at different levels with different partial pressures. So once I have partial pressures that get up high enough, like in the lung, I am going to fully saturate hemoglobin. And again, when I go through the tissues, that's resting tissue now, not exercising, I'm going to still be 75% saturated. And then if I do exercise, I'm still 25% saturated, or close to it. If it's vigorous exercise, it'll go a little bit lower. And but you see, it's pretty close to that. So what that means is we have a little bit of a buffer. So as we start to exercise more and more and more, we still have enough oxygen available for our muscles and our brain and every other tissue that needs it. So any kind of temperature increase, pH decrease, which would be hydrogen ion increase, partial pressure, CO2, and bisphosphoglycerate levels, all of those can lead to differences in hemoglobin saturation. And it modifies the protein, and it makes it where it doesn't want to bind to oxygen as well. So it decreases what we call the affinity. And this can occur in the systemic capillaries and enhance oxygen unloading, and it shifts it. So again, as we have, metabolic processes that change the chemistry of the body, the hemoglobin is going to change in response to that. So we can see this on a graph. So again, if we look at normal body temperature, we can see that if we go higher, the curve shifts to the right. As our body temperature goes lower, we shift to the left. So again, I talked about this with trout a minute ago and fish, that the water they live in can contain more oxygen than slow-moving, stagnant, or warmer water because it can hold more gas. And we have to look at the saturation curve here based on that same solubility principle. Same thing happens with pH. We can change the pH, and basically we can. Excuse me, I'm sorry. This is still the oxygen level. There's another one with pH on it. Not even paying attention to it. The pH here, you can see changes from 7.4 to 7.2. This is also going to change the partial pressure of oxygen. So 7.6, 7.4, 7.2, you can see this is going to shift back to the right. I was thinking there's still temperature, but it's not. It's a pH scale. But basically, you're just seeing shifts in the amount of saturation or the saturation point of hemoglobin with varying temperatures and varying pH levels. Right? So when cells metabolize glucose, the partial pressure of CO2, of course, is going to increase. They're burning glucose and producing CO2 as a byproduct. And then as a result of that, hydrogen ions also increase in concentration. So that weakens the bond of hemoglobin to oxygen. We call that the Bohr effect. And then the heat production increases also can directly and indirectly decrease the hemoglobin affinity for oxygen. So again, when you get a fever, it's not just the enzymes and things that aren't working properly, but you may not be able to get enough oxygen. So let's look at COVID-19, for example. One of the biggest underlying factors in how you survive this disease is whether your immune system, number one, gives you what's called a cytokine burst and attacks your lung cells. But the other one is also the temperature, because when you increase the temperature and your immune system's attacking your lung cells, now you have a double whammy. You have the lung cells not being able to function properly and get oxygen in the blood, but you also, on the other side of that, you have the lung being destroyed by your immune system, and that double whammy makes your lung not function, and you're not getting enough oxygen in the blood. Well, if you're not, your heart then is going to suffer. You start having heart trouble and organ trouble, and then eventually you die. And that's why people with comorbidities and underlying conditions have much more problem with this. If you have emphysema or you have heart issues or blood pressure issues, you're much more likely to die from COVID, because it's not the disease itself that kills you, it's the factors that go along with it that kills you. It changes your metabolic activity in your body, and it can cause a lot of different problems with breathing and getting oxygen in the bloodstream, and again, that leads to organ failure and so on. Right. So when we look at a homeostatic imbalance, again, we can talk about COVID here too, because this is exactly what it does. It basically takes the ability of the body to get oxygen into the blood away, damaging the lung tissue. It's not the virus that damages it, it's your immune system and your immune response to that virus affecting those cells that causes that to happen. So anytime you hear hypoxia, that just means inadequate oxygen delivery to tissues. And there's a lot of causes of this. You could have too few red blood cells. That could be from blood loss, donating blood, your bone marrow is not working properly, et cetera, et cetera. Abnormal hemoglobin, that could be a genetic factor. Blocked circulation, that can be plaque buildup, that can be clots. Metabolic poison, there's things like snake venom and things that can cause metabolic issues. Pulmonary disease, you have COPD, emphysema, bronchitis, lung cancer, all of those things can lead to hypoxia. And then carbon monoxide is it's kind of a silent killer. Most people don't pay attention to it because it's powerless and odorless, and you never know what's around, and you just basically fall asleep and die. So if you have anything that burns fuel, whether it be natural gas or wood or pretty much anything that burns gasoline, you don't want that building up in a building. You know, if you, let's say, run a generator in a garage and you have the doors closed, you're filling it with carbon monoxide. That's just not a good idea because that can lead to that. All right, now CO2 is a different animal. It's going to be transported in blood in three forms. 7 to 10% dissolved in plasma. 20% is bound to the hemoglobin. We call that carbaminohemoglobin. The vast majority is going to be transported as the bicarbonate ion. And this involves a chemical reaction. And I want you to know these two diagrams I'm going to show you in a minute for your test. I want you to memorize them. I don't want you just turning them out and having them in front of you. You need to know what all of this stuff means on this. So I want you to study it. Okay? I'm not going to ask you a direct question about it. I'm going to ask you, why would this occur if this happened? Okay? So definitely make sure you study these when I show them to you, and I'll make note of it when I get there. Right? This is the reaction you need to know. CO2 and water form carbonic acid. Carbonic acid dissociates into hydrogen ion and the bicarbonate ion. Bicarbonate, over here on the right, is the one that is going to be responsible for carrying the majority of the CO2 in the blood. And what we can do is carry it in the blood by taking CO2 from the cell, combining it with water, making carbonic acid, adding a few hydrogen ions. And we have some other buffers besides this one that will neutralize those. We also produce this bicarbonate ion. When we get to the lung, we reverse this. We take bicarbonate, add it to hydrogen, convert it back to carbonic acid, to water, to carbon dioxide. These are the two vapors that we breathe out every time. So this reaction is going to be very important in understanding how blood is going to deal with oxygen and CO2 and how these things move in and out. Again, if you know both of these diagrams I'm about to show you, you'll be in good shape. They're opposites of each other. So if you learn one, you'll know the other. Okay? So let me move over to here and let's just kind of explain this in a way that's easy to understand with a visual type of presentation here. So give me one second. Let me get some water. Good. All right. So anyway, what we're looking at here is the release of oxygen and carbon dioxide pickup at the tissue level. So this is our red blood cell over here, the oblong egg-shaped cell that you see. It's kind of biconcave, as we talked about back in the blood chapter. These are our tissue cells. Remember, these are big factories. And all they're doing is taking oxygen and glucose and converting it over into energy, and the byproduct is CO2. And we need to get rid of it. So this is the hard one to kind of understand because CO2 can go in so many different pathways to get to the same result. Ooh, excuse me. So the CO2, a little bit of it's going to dissolve in the plasma. It's not very soluble here, but it's easy for it to get into here. So only a small amount of it's going to go into the plasma. CO2 can also go through what we call the slow reaction and form bicarbonate ions. And in that, it's going to produce a hydrogen ion that binds to plasma proteins. In order to counteract that positive ion leaving, we have the chloride shift where the negative ion comes in and balances the charge back out. Right? So that's kind of some of the chemistry involved. Now, if that was the only way that CO2 could be pulled out of cells and put into the blood, it would be super inefficient. Right? So what we need to look at, more importantly, is the reactions that we have. This is where our enzymes come into play. So carbonic anhydrase. What does that mean? It's taking and forming carbonic acid, and anhydrase means it's taking a hydrogen away. So we're going to take CO2 and water. It's going to go through what we call the fast reaction. This is the same one as the slow one up here. Except this is going to have an enzyme to catalyze it. This happens very rapidly. We produce the bicarbonate ion, and then we basically can pump that out and then pump a chloride ion back in to balance all the charges out so that we don't lose charge inside the red blood cell. Again, concentration gradients, charge gradients are very, very important in physiology and understanding how this works. Right? Now, the other way CO2 can come in is by binding to hemoglobin and becoming carboaminohemoglobin. And that's going to also be carried in the red blood cell. Now, if you understand this, you will understand the next diagram that I'm showing you, because it's the complete opposite. What happens at the lung instead of at the tissue? So at the lung, the exact opposite reactions happen. So now the CO2 dissolved in plasma goes back into the alveoli. The slow reaction occurs, converts that back to CO2, and it gets put into the alveoli. Fast reaction happens, and it gets converted to CO2 and goes back in the alveoli. And then finally, we take the carboaminohemoglobin, and that is converted back to CO2 that goes into the alveoli. So now we have this blood cell that's dumping off CO2. That makes the affinity for oxygen increase, or it makes it want to bind to oxygen even more. And the vast majority will come in and bind to that hemoglobin that was left over from the reaction up here. Now we will form oxyhemoglobin that will go throughout the body and provide the tissues the oxygen that they need. Make sure you memorize these two diagrams. Again, they're both the same diagram with the arrows going in the opposite direction. One is at the lung here, and this one is at the tissue level. It's a complete opposite reaction. So it's really pretty simple. If you know how it goes in one way to the lung, you know how it comes out the other way to the alveoli, and vice versa. Right? So hopefully that will make sense to you, and be able to kind of figure out how CO2 and oxygen are both carried. Now, the Haldane effect, really, this is just based on the concentration of oxygen in the atmosphere. CO2 is going to always want to bind to hemoglobin more readily when the hemoglobin doesn't have oxygen. This can cause a problem. If you're in high altitude situations, there's not much oxygen up there to begin with, but you are still producing CO2 as a byproduct of your cellular respiration, as long as you have energy and you're able to breathe at least a little bit. So this is the Haldane effect, and we see that with people that climb tall mountains like Everest and some of the others that get up 29, 30,000 feet. And I think Mount Everest, the highest mountain on terrestrial land masses, I think it's 29,000 something feet, so almost 30,000 feet. No one can breathe up there. And it's because of the Haldane effect. I'm sorry, yawning. It's getting late. But at the tissue, more carbon dioxide enters the blood, and then more oxygen is going to enter the tissue. And this is going to eventually equal out, and then the red blood cell is going to move on. Now, this sounds like it happens over a 2-minute time. This happens in less than a millisecond that these gases are exchanged. So I'm going to keep that in mind with that. We've talked about CO2 and blood pH. I gave you the demonstration where I blew in the little jar the other day and showed that CO2 converts to an acid, carbonic acid, when you're breathing in that. So I'm not going to go over that very much. And I'm going to stop here and then finish out this tomorrow. I am tired and my voice is about to go out, and I have a lab tomorrow that I need to prepare for. So anyway, I'll start out with control of respiration. We'll get into the brain functions and how that controls respiration. And hopefully I can get the digestive videos up tomorrow and get those online for you. Right?